Your nasal microbiome is just as important as your gut microbiome in its effect on health and disease, yet it's just now really starting to be researched. Watch this video if you'd like to learn more about how it affects your health.
What up, fam? So today I wanted to talk about something that is a really big deal and has a lot of new research emerging on it, but is not necessarily very well understood yet. And there's very little awareness about this within the general public and also probably within the medical community, at least Western medicine. So we all know it takes about 10 years for the research to really catch up in clinical practice, or I guess you could say it the other way around. Doctors don't really know about the current research as it's being published. So you may be suffering from some sort of chronic illness, or you may just have a lot of inflammation that could later turn into chronic illness, or you may just have something like a post-nasal drip that's kind of annoying, but doesn't seem to be causing you, many systemic problems. Either way, it's going to be important for people to start hearing about and getting familiar with the sino-nasal microbiome. So what is that? Well, everyone's probably heard of the gut microbiome, which is, as you probably know, the bacterial colonies basically in your digestive tract, or more specifically that would be in, like your intestines, your large intestines. So the sino-nasal microbiome is the microbiome of your nose. And that's your nasal cavities as well as your sinus. And there are, different regions within your sino-nasal cavity. And they may have, a different, I guess, pattern, or a different, diversity of organisms. And the microbiome is a collection of all of the microbes. So it's not just bacteria, but it's bacteria, viruses, fungus, fungi. So it's everything. And there's a lot of research happening on the gut microbiome and how that impacts health, as well as, obesity. And we're actually learning a lot quicker about the sino-nasal cavity in research. And I think what we're learning there can teach us a lot about how we're researching the gut microbiome. So I have notes because I don't want to leave anything important out. So, okay, does the bacteria that's in your nasal microbiome mirror the bacteria that's in your gut microbiome? That was kind of my first question, and the answer is no. The general taxa of the bacteria is the same. So you have Firmicutes and Bacteroidetes and basically all of the same types of bacteria, but the percentages in which they exist in those two locations are completely different when you compare within the same person. So if you're thinking, well, disease is caused by, this type of bacteria being in overabundance, well, the location matters, and it's not as black and white as that. Nothing is black and white in nature. So I'll talk about that again at the end of the video, but just so you know, they're not the same. The microbiomes are different, even, in the same person. So, okay, why should you care about this? Well, disease is linked to the microbiome of your gut and to your sino-nasal cavity. And just inflammation in general is linked to the diversity of the microbiome in these two locations. So some research is saying that the more diverse the nasal microbiome is, the less inflammation you have. And some research is saying that diversity doesn't really matter. It's more about the quantity of quote-unquote bad bacteria that's colonized in the microbiome. So the research isn't fully there yet because there are some limitations to the research, but all of the research agrees that the microbiome can indicate disease and inflammation. And one of the main things that indicates inflammation and disease is the presence of staph. And there's different types of staph. So the two pathogenic types that are most commonly studied in the nasal cavity are MRSA, which is methicillin-resistant Staphylococcus aureus, and MARCoNS, which is multiple antibiotic-resistant coagulase-negative staph. So if either of these two types of staph are the highest concentration of bacteria in your nasal microbiome, then you're probably going to have problems. High concentrations of those two types of staph are correlated with inflammation and disease, even systemic inflammation, not just localized inflammation. So what are factors that influence the sino-nasal microbiome? Well, history of smoking cigarettes, and not just the history of it, but actually, currently smoking cigarettes as well. But there was one study that showed if you've ever smoked cigarettes in your life, not just tried it, but ever habitually smoked cigarettes, then you are predisposed to having, these two types of staph in your nasal microbiome. And you're also predisposed to systemic inflammation. And then a history of other inhaled irritants would affect the nasal microbiome. So pollution in the air, if you live in China, like my little brother, then this is going to affect your nasal microbiome. And also mold. So if you know anything about me, you know that I have suffered from mold illness, I've healed myself, I've been exposed again, and I'm in the process of healing again. And, mold produces mycotoxins, which are among the most carcinogenic substances known to man. In fact, the most carcinogenic substance known to man is a mycotoxin. And this happens indoors, when you have water damage, mold and other bacteria proliferate on the building materials if the water damage is not corrected quickly enough, if it doesn't dry fast enough, or if the building materials aren't, cut out and removed from the building. So this has been shown in the studies. Breathing in indoor mold has been shown in the studies to alter the sino-nasal microbiome in a negative way. And I'll talk more about that in the video as well. Also, a history of asthma has been shown to have some sort of effect on the nasal microbiome, or at least be associated with negative changes in the nasal microbiome. So we don't really know which comes first, the asthma or the changes in the microbiome. But back to mold illness, there has actually been a significant number of studies done. I think at least one by the EPA was done in California, and they found that children with a history of asthma were living in homes that were colonized by a type of mold called Cladosporium. And it's a fungus that kind of looks like black mold. It's not as, the mycotoxins it produces are not as pathogenic as black mold, but the association was there. So this type of mold is associated with asthma. So again, these are just associations that, you know, a history of asthma is associated with negative changes in the nasal microbiome. Well, it could all come back to inhaled irritants like mold. So what does not affect the sinonasal microbiome? This is good to know because, you know, for research purposes and for practical purposes, topical saline does not have an effect on the nasal microbiome, at least not a significant effect. So if you use a nasal wash, you're not doing anything negative to your microbiome. You're probably not doing anything positive directly either, but maybe indirectly if you've been exposed to an inhaled irritant and you're using, you know, something to flush it out. Other things that don't seem to affect the nasal microbiome, the presence of nasal polyps. Pre-op or post-op, these don't seem to have any association with the actual diversity of your microbiome. And using nasal steroids like topically doesn't seem to have any effect either. I don't know, go figure. So as promised, I want to talk about how the limitations of sinonasal microbiome research can shed light on, you know, how we understand the gut microbiome. So the limitations of the current research for the sinonasal microbiome are pretty extensive because it's in its infancy. So the presence of biofilms, which I haven't mentioned yet, but certain bacteria lay down or participate in the creation of biofilms. So this is a film that basically protects the bacteria from things like antibiotics, which makes stuff like MRSA and MARC-ONS really tough to treat because you can't just throw an antibiotic in there. You can't just take an antibiotic orally. You have to break up that biofilm. And there are some things that have been proven effective to do that and some things proven ineffective to do that. But if the biofilm is there to protect the bacteria, it's going to be hard to sample that bacteria as well without breaking the biofilm. So that's one limitation in research, is that we have to figure out exactly how biofilms are impacting the samples and the results. Also, the types of bacteria that make biofilms are difficult to culture. So these bacteria, which are essentially the focus of you know, the study of how pathogenic bacteria in the microbiome affect health, are in and of themselves elusive, really hard to culture. So that's tricky. Also, sampling techniques, and this is something that all of these things apply to the gut microbiome as well. So people will, you know, do research on feces. If it's a rodent study, I'm sure they could, you know, cut open the intestines, you know, sacrifice the animals and cut open the intestines. Well, in humans, we can't really do that. So, you know, the location that you're sampling from has an impact on the microbiome. So whether you're deep in the sinuses or, you know, the nasal cavity, like these areas have a different population of bacteria even within the same person. So for the sinonasal cavity, you'll see some research where they use lavage, and that's basically like a rinse where they're trying to capture bacteria. Well, you have to consider they're diluting the bacteria. There's no way to tell what part of the nasal cavity it's coming from. And you may even have contamination from the skin microbiome. So these are things that apply to the gut microbiome as well. We don't really know what we're doing here, so it's hard to really make any, you know, to really extrapolate anything concrete. We're just kind of figuring it out along the way. So other things that we need to take into consideration when we're studying these microbiomes is that it's not just the bacteria and it's not just the biofilms that matter. It's actually stuff that the bacteria produce and how biologically active those things are and how well we can study them. So some of the research in the sino-nasal cavity microbiome has looked at something called extracellular vesicles, which contain biologically active components of the bacteria. So things like bioactive lipids, proteins, and nucleic acids. Nucleic acids, that's DNA. So these things can be delivered to your cells and they come from the bacteria. So these things can modulate how your cells are working. And so it's not just the bacteria, it's, are the bacteria that are present producing these extracellular vesicles or EVs, and what's the impact of the EVs on your cells? So is even studying the bacteria itself that helpful? So we need to focus more on stuff like that, basically like the functional impacts of the bacteria, not just the bacteria, because the limitations on studying the bacteria haven't yet been overcome. So other things that can affect the research, were antimicrobials like antibiotics or something like that used around the time of the sample collection, because that will affect the sample. The age of the person, so we know that the gut microbiome changes as we age naturally. So how does the sino-nasal cavity change as we age? And are researchers controlling for age when they're doing this research? Not always, so that's important. And then the severity of the participant's disease. So some of this research is done in, populations that have systemic inflammation or localized inflammation in the nasal cavity, and they're not necessarily controlling for how severe that person's disease is. So you're getting results in this research of people, at different places in the disease progress, basically. So, other important factors to consider. I mentioned before that inhaled irritants have been associated with the presence of these two, pathogenic staph bacteria. Well, which comes first? Did the person get colonized with Marcons or MRSA, and then that led to inflammation that caused disease? Or did inflammation from the inhaled irritants cause the mucosal lining of the microbiome to become infiltrated? So that's something that we don't fully understand, and it's possible that both happen simultaneously, and that leads to disease. In nature, nothing's black and white. So a little bit of both could be happening at the same time. Since we know that people can have MRSA as well as Marcons in their nasal cavities and be perfectly healthy with no signs of systemic or local inflammation, we can kind of figure out some things from there. So what is the turning point? What makes the presence of these bacteria become a problem? We don't really know that, but we do know that, there is one major connection between the sino-nasal microbiome and the gut microbiome. And that is, as it relates to disease, at least. And that is that we can't be colonized by these pathogenic bacteria And we can't have systemic inflammation caused by these pathogenic bacteria without our mucosal membranes and other barriers in the body being compromised. So your blood-brain barrier has to be compromised in order for there to be neurological inflammation. Excuse me. Your gut lining has to be compromised in order for there to be systemic inflammation. Your sino-nasal microbiome or mucosal lining has to be compromised in order for there to be inflammation. So what are the factors that cause your mucosal linings and these protective barriers in the body to become or to lose their integrity, basically? One of them is actually genetic. So you can have a genetic defect on genes that code for interleukin 22, I believe. Yeah, IL-22. This is a cytokine. All the interleukins are cytokines. Some are inflammatory, some are anti-inflammatory. And IL-22 is actually associated with the permeability of these mucosal membranes of your gut lining and your nasal cavity. And you can have just a genetic defect that predisposes you to having these issues. But there are also blood biomarkers that are associated with this as well. So if you've been exposed to a moldy environment, like a water damaged building, then there are some biomarkers that you can measure in the blood that will either become high or become low, and this can precipitate that type of change in the mucosal lining. So TGF beta 1, transforming growth factor beta 1, this is one that I monitor in myself, and it can become high in a number of different inflammatory processes, including exposure to mold mycotoxins and biotoxin illness. And TGF beta 1, when it's high, activates pro-inflammatory Th17 cells, so these are immune cells, that secrete cytokines such as IL-22. And IL-22 can decrease the integrity of the barrier of your intestine, your brain, your nasal mucosa. So that's one thing. Other things that I also monitor that can become altered when being exposed to something like mold would be high MMP9. So that's matrix metalloprotein 9. And these are kind of highly technical, so your doctor is probably not going to know what these are. If you want more information about this, just reach out to me. But MMP9 has to do with the integrity of your connective tissues in general, but also specifically the connective tissues that make up your gut and your nasal cavity. MMP9, when it's high, is associated with aortic aneurysms. It's associated with genetic connective tissue disorders. It just has to do with collagen production. And it will get high in certain people that are exposed to mold. And we know that there's also a positive correlation between these biomarkers becoming altered and the presence of nasal mark-ons. So we don't know which makes you susceptible to which, or if it happens simultaneously. But the presence of multi-antibiotic resistant coagulase negative staph in your nasal cavity is related to these biomarkers becoming, high or low, abnormal, basically. And then low MSH, low melanocyte stimulating hormone. This can also lead to, issues with the integrity of, these barriers within the body, and is also associated with multi-antibiotic resistant coagulase negative staph. Okay, so let's get down to what everyone wants to know, which is, how do you affect or how do you address the health of your sino-nasal microbiome? The truth is we don't fully know all of the ways yet that there are very few products designed at increasing the diversity of the beneficial bacteria in your nasal cavity. It's not as easy as just taking a probiotic. That's what a lot of doctors recommend for the gut microbiome. Honestly, that's probably not as easy as just taking a probiotic either. In my opinion, it's definitely not. These are complicated things. So, first of all, we need more research. That's the first thing we need to do to know how to address the sino-nasal microbiome. We need to do more research. The second thing we need to do is test. Test for MRSA, test for Marcons. If you're a person with chronic illness or inflammation or any unexplained health complaints, or maybe even explained health complaints but you're not getting better, test these things. Have your doctor test them. You don't have to have your doctor test them, actually. You can test some of these yourself. And I can give you information to do that. You order a Q-tip, swab your nose, mail it into a lab, and they'll tell you what is in your nose, and they'll also tell you what antibiotics it will respond to, because these things are resistant to antibiotics. That's an important takeaway, is that you can't just take an oral antibiotic and hope that these are going to go away. So after testing, you need to remove yourself from exposure to inhalant irritants. So if you're exposed to mold, you need to get out. And that's easier said than done. I can help you do that. I've helped a lot of people do that. I've done it myself several times. It's complicated and it's hard, but you can do it, and you have to do it. That's it. You have to do it. We know that what we breathe in changes our nasal microbiome. It also changes our gut microbiome. It causes systemic health problems as a result. So take the first step, reach out to me, we'll talk about it. So after you've removed yourself from exposure, what do you do to address the gut or the sino-nasal microbiome? You have to break the biofilm if there's pathogenic bacteria that have produced a biofilm. And there are some ways to do this that are more effective than others. Xylitol nasal spray from birch trees can break biofilms. EDTA can break biofilms. EDTA is a chemical used actually as a preservative in a lot of things, including a lot of foods, but it's also used in chelation therapy to remove certain positively charged ions from your body, like toxic heavy metals like mercury and lead. Those are the two probably most effective biofilm breakers. Okay, then antimicrobial treatment. Once you know what antibiotics these pathogenic bacteria are sensitive to, you could go that route. That's actually not been proven that efficacious though. You might want to experiment with other antimicrobials like I'm doing. And I don't recommend that anybody do this at home, but I'm currently trying to hack my nasal Marcons, and I'm doing it with ozone therapy, among other things. So another way to affect your nasal microbiome in a positive way is to breathe healthy outdoor air. Go to the beach, go to the woods. Don't go to a beach that has red tide or blue-green algae. Get out of pollution for sure, but breathe in the outdoor air, because that's been shown in the research to be effective in increasing the diversity of beneficial bacteria in your nasal microbiome, also in your gut microbiome. And also be around animals. The research shows that breathing pet and animal dander can actually increase the beneficial bacteria in your sino-nasal cavity. And you don't want to kiss any pets. Dogs can harbor mark-ons in their nose and they will give it to you. And if you've worked really hard to get rid of your mark-ons and then you get it from a dog, you're going to be really upset with yourself. So don't kiss your dog, but have a dog or play with a dog, whatever. Another study showed that being around farm animals can be beneficial. So the dander from farm animals. Yeah, I don't know, who knew? And then the last thing is VIP nasal spray. I hesitate to even say this because the researcher who's doing all of the work on VIP nasal spray doesn't really want doctors using it yet, because there aren't enough doctors that really understand how to use it, and he doesn't want it to be used incorrectly and then people get sick, and then it gets made off limits for everyone. So, don't just jump head first into this, but I've done my research, I know what I'm doing, and I'm using VIP nasal spray diluted by like a 1:10 ratio with preservative-free sterile saline. And VIP stands for vasoactive intestinal peptide. And this is something your body produces naturally. It can get low if you have nasal colonization of these pathogenic types of staph. And we're not sure if low VIP leads to colonization or if colonization leads to low VIP, or if they happen simultaneously. Again, it's not a black and white thing. The research is not there, and it won't be there anytime soon. So we do know that if you supplement with a very low dose of prescription VIP nasal spray, it can help restore the integrity of these mucosal linings, and it can, in general, just help you heal. It takes a little bit of the burden off of your body to heal itself. So if you want to know more about that, reach out to me. Don't just go to your doctor and ask for it. It's a compounded prescription. It's very expensive. It's like $200 a month. So don't take this lightly. I don't recommend doing it. I don't even recommend bringing it up with your doctor. But if you want some more information on it, I got you. So the last point I want to make is follow me on Instagram to watch me biohack my mark-ons. I do live videos, I do pre-recorded videos of me biohacking my mark-ons with nasal ozone and other things as well. I talk a little bit about VIP there too. And you can also just follow my journey in general. If you have any health issues that are unexplained, or if maybe your doctor has an explanation but you're not getting any better, follow me, ask me, questions, reach out to me. If you're not at the point where you're ready to reach out, just watch, and see what I'm doing, what I've gone through, where I'm going, this sort of thing. That's how I learned a lot, was just watching other people that were dealing with the same thing. And go to my blog, experimentalbetty.com, and you can read articles about this stuff so that you can have it, saved to your computer for, a resource. And I usually cite the things that I write with scientific research. Everything that I've said today came from the research, and I'm going to write a post about this and link it for y'all so you can see it. Some people prefer to read and some people just prefer to have it. That's it. Thank you for watching, and like I said, always feel free to reach out to me and I can give you more information on my journey, and it may help you. You never know.